Look around this microbiology lab. Then look down to see the microscope slide colonized by bacteria. Bacteria exist in protective structures called biofilms, which can be found on most surfaces. Click the trigger on your handset to continue. You are now looking at a biofilm. It was created using a bacteria called Pseudomonas aeruginosa. This type of bacteria is an expert in forming biofilms. It lives in soil and water, but can also cause severe infections. Biofilms form when a bacteria lands on a surface and sticks to it using specialized surface molecules. That bacteria then grows and divides, and other bacteria are attracted to it. These clusters of bacteria secrete sugars known as exopolysaccharides, which form a protective matrix around the cells. The matrix grows to create a dynamic structure that can support many different microbes. Notice the flow of liquid around the biofilm. Fluid channels are bringing fresh nutrients in and carrying toxic waste away from the colonized cells. Different concentrations of nutrients and oxygen exist throughout the biofilm, allowing diverse communities of bacteria to coexist. The bacteria in this biofilm experiment has been labelled with fluorescent protein, which makes them glow bright green. Once the biofilm formed, it was treated with a red dye that binds it to the DNA of dead cells, so dead bacteria can appear red. This experiment was set up to investigate how the biofilm responds to different doses of antibiotics. Biofilms are hardy and difficult to shift. In the body, this is not an issue, so long as your microbiome is healthy, but it can be a problem when trying to get rid of an infection. Antibiotics can diffuse through the matrix very slowly, and the bacteria within can be highly resistant. Now, let's see what's happened. We're going to go down into the biofilm and test out 3 different antibiotics to see how effective they are at killing the bacteria. Now, when you're ready, use the trigger on your handset to begin. let's try out the first of our antibiotics, gentamicin. This one has a broad spectrum of activity. Point the controller at the icon on the right, and use the trigger to apply a dose and see what happens. Okay, you've applied a fair dose of gentamicin. As you can see, lots of bacteria have turned yellow and red, meaning they are dead. However, if you keep watching, you will see the biofilm heal and turn green again. Some bacteria have tolerated the dose and continue to grow. You should apply more doses to make sure all the bacteria in the biofilm are completely dead. Well done! You have used gentamicin appropriately to defeat all the bacteria in this biofilm. Now, let's try a different antibiotic and see how well it does. Use the trigger on the controller to continue. Now, let's try penicillin. This is one of the earliest antibiotics discovered. It attacks the bacterial cell wall. As before, to apply a dose, point your controller at the button on the right and use the trigger. Oh no! Penicillin doesn't seem to have had much of an effect at all. Many types of bacteria are already resistant to certain antibiotics. Maybe if you apply enough doses, you might defeat the biofilm. Well, it took a lot of doses, but you managed to defeat the biofilm with penicillin. There is just one more of our antibiotics to try out. Use the trigger on the controller to continue. Our final test is ciprofloxacin. This antibiotic attacks bacterial DNA. Let's see how many doses it takes for this one to wipe out the biofilm. Point the controller at the button on the right and use the trigger to apply some doses. That's good, keep going. And you've done it again. The biofilm is totally dead thanks to your efforts. And with a little help from ciprofloxacin. When people take antibiotics to treat an infection, they must finish the full course. Otherwise, the surviving bacteria can develop resistance to whichever antibiotics was used. Established biofilm infections can be very difficult to treat because the antibiotics take a long time to travel through the matrix, and the infectious bacteria have become more and more resistant. This is the case for many individuals with cystic fibrosis, who suffer from chronic lung infection by bacteria like Pseudomonas aeruginosa. A better understanding of bacterial biofilms and how they respond to antibiotics will help researchers to developing new treatment strategies. This virtual reality biofilm tour was created by microbiologists and computer scientists working together at the University of Salford to help with their collaborators at the universities of Louisville and Liverpool and support from LabTech and Fluxium. Thanks for playing at Microbiome. Please remove your headset. Goodbye.